Welcome to the lecture series on cell communication. I'm Janine Post of the Developmental Bioengineering Group of the University of Twente. In this lecture, I will discuss additional regulatory principles and methods for investigating cell signaling effects. This lecture is the last of 5 lectures on cellular signaling. The other lectures can be found on my YouTube channel. Illustrations in this video are based on the book Essential Cell Biology and from my previous work and from literature found via PubMed. Links to the literature can be found on the last slide. In lectures 1 to 4 of the cell communication series, I discussed changes of activity of signaling proteins by GTP-GDP in the case of G protein coupled receptors, or phosphorylation of the target protein by protein kinases. Another regulatory principle is binding of small ions to proteins, thereby changing the conformation of the protein and subsequently its activity. The best known example is calmodulin, which is present in the cytosol of all eukaryotic cells. Calmodulin has a dumbbell shape with 2 globular ends connected by a long alpha helix. Each of the globular ends has 2 calcium binding sites. Upon calcium binding, the conformational change of the protein allows it to interact with a wide range of target proteins, thereby altering their activities. One class of calcium-dependent proteins is the calcium calmodulin-dependent protein kinases, or chemkinases. Chemkinases are able to influence other processes in the cell by phosphorylating other proteins. Among other functions, chemkinases are found to be important in some forms of learning and memory. Second messengers, like cyclic AMP and calcium, are hydrophilic molecules that generally act in the same cell where they are produced. Some other molecules are small enough or hydrophobic enough to pass across the membrane and carry a signal directly to neighboring cells. An important example of this is the gas nitric oxide. The gas diffuses rapidly from its sites of synthesis and slips into neighboring cells. However, the distance it travels is limited due to its reaction with oxygen and water in the extracellular environment, which converts NO into nitrates and nitrites in seconds. The neurotransmitter acetylcholine causes the blood vessel to dilate by binding to a GPCR on the surface of endothelial cells, thereby activating a G protein, GQ, to trigger calcium release. In turn, calcium activates nitric oxide synthase, or NOS, stimulating the production of NO. Nitric oxide then diffuses out of the endothelial cells and into adjacent smooth muscle cells, where it regulates the activity of specific proteins, causing the muscle cells to relax, thus increasing blood flow. One key target protein that can be activated by nitric oxide in smooth muscle cells is guanylyl cyclase, which catalyzes the production of cyclic GMP from GTP. Cyclic GMP is degraded by phosphodiesterase 5, or PDE5, forming GMP, and as a result the smooth muscle cell contracts again and blood flow is reduced. One well-known drug that targets this particular pathway is Viagra, which prevents the breakdown of cyclic GMP, resulting in a prolonged signal, a longer increase in blood flow into the spongy erectile tissue of the penis, and thus prolonged erection. Note that nitric oxide gas is highly toxic when inhaled and should not be confused with nitrous oxide, also known as laughing gas. In this example of contact-dependent signaling, a Notch receptor on the cell surface can interact with ligand delta on an adjacent cell. Delta binding to Notch activates the receptor, which is then cleaved, and its cytosolic tail is free to move to the nucleus, where it acts as a transcription regulator. This type of contact-dependent signaling is found in tissue development and tissue patterning. Some small hydrophobic hormones bind to intracellular receptors that act as transcription regulators. Take, for example, cortisol. Cortisol is one of the hormones produced by the adrenal glands in response to stress. It crosses the plasma membrane of a target cell and binds to its receptor protein, which is located in the cytosol. The receptor hormone complex is then transported into the nucleus via the nuclear pores. Cortisol binding activates the receptor protein, which is then able to bind to specific regulatory sequences in the DNA and activates or represses the transcription of specific target genes. Whereas the receptors for cortisol and some other steroid hormones are located in the cytosol, those for other steroid hormones and for thyroid hormones are already bound to DNA in the nucleus, even when hormone is absent. Intracellular signaling pathways are mapped out using many different experiments. For example, a receptor could be isolated using another protein as bait. For example, the insulin receptor was isolated using a chromatography column that had insulin attached to it. Other proteins binding to the receptor could be isolated using co-immunoprecipitation, by which you use an antibody against one protein to pull out the other proteins that are bound to it. Mutations of the DNA encoding for the protein can help to identify the order of proteins in a signaling pathway. When determining the order of proteins in a pathway, one can use cell lines that each carry a mutated form of one of the proteins in the cascade. Consider a pathway consisting of a receptor tyrosine kinase, a protein X, a protein Y, and RAS. You want to investigate if RAS is upstream or downstream of protein X. If RAS is upstream of protein X, then inserting a overactive form of RAS in a cell line with a defective protein X, cell line XMOD, will not rescue the signal. However, if RAS is downstream of protein X, then this will rescue the signal. A similar experiment can be done with a cell line containing a defective protein Y. In this case, addition of RAS to the YMOD cells did not restore the signaling activity, indicating RAS is upstream of protein Y. Based on these results, the deduced order of the signaling pathway is shown. Mutations of the DNA encoding for the protein can help to identify where protein interactions take place. To determine which tyrosine binds to a specific intracellular signaling protein, a series of mutant receptors is constructed. And the mutants shown, tyrosines tier 2 or tier 3, have been replaced, one at a time, by phenylalanine, shown in red, thereby preventing phosphorylation at that site. As a result, the mutant receptors no longer bind to one of the intracellular signaling proteins. The loss of binding of the intracellular protein, or the cell's response, can then be used to determine which protein binds to that site. It is important that the mutant receptor is tested in a cell that does not have its own normal receptors for the signaling molecule. Research can be done on different parts of the signaling pathway, on ligands, on receptors, on intracellular signaling molecules, target proteins, and other targets, such as mRNA expression, cell migration, and metabolic changes. At the level of ligands, one can ask numerous questions. For example, looking at the ligand-producing cell, the expression levels at the mRNA and protein level may be interesting to look at. Also interesting is to investigate how the ligand binds to the receptor and what concentration is needed to activate a target cell. In addition, ligands may be used to track ligand-receptor complexes, as we have shown before, for different EGFR types and the ligand EGF. In this study, we showed that ligands bind to receptors located in the membrane of Filopodia. and that the receptor complex travels to the cell body and internalizes. Receptors are important molecules, and many mutations are linked to pathologies, including cancer. Here you see an example of a crystal structure of EGFR on the left. And on the right, you can see how two EGF molecules are bound to two molecules of EGFR. Crystal structures can serve as a basis for mathematical modeling of the effects of mutations of ligands or receptors on structure and thus function of the receptors. In the case of intracellular molecules and target proteins, one would use methods that can identify specific proteins. These methods are often based on using specific antibodies against the protein of interest. Protein expression can be investigated using Western blot and immunofluorescence. In addition to concentrations, immunofluorescence can tell you something about the localization of the protein in the cell. In this example here, we stained the nucleus with a specific DNA dye and used two specific antibodies, in green against CD31 and in red against integrin beta, to show the membrane staining of these proteins. One could also use a fluorescent protein directly coupled to a protein of interest, or a protein located in the organelle or structure of interest. This example, in which we used a GFP fusion of a membrane protein, shows that binding of a ligand, in red, to the receptor immediately changes the cytoskeletal activity, as can be seen by the extensive membrane ruffling. Again, we can see that the receptor-ligand complex is quickly internalized into the cell body. Target gene expression can be assessed by qPCR, RNA sequencing, or microarray assays. In this example, we show that in qPCR you can quantify the amount of RNA originally present. Cell migration can be visualized using cell tracking or by a scratch assay, as shown here. Metabolic changes are often investigated using mass spectrometry. One can ask many research questions using a toolbox of fairly limited number of tools. For example, one can investigate protein function by using antibodies, fusion proteins with, for example, green fluorescent protein, or specific dyes. This allows you to look at a number of mechanisms. Take, for example, the use of antibodies. You could use a protein-specific antibody in many different tools, such as Western blot, co-immunoprecipitation, immunofluorescence, ELISA, and many more. Tools based on fusion proteins can be used to look at protein mobility, localization, and co-localization, but can also be used to fish for protein interactions if a specific antibody is not available. Dyes are often used in histology, but also in live cells. For example, membrane dyes can be used to look at cell migration in a tissue. Another important tool in the box are small oligonucleotides, composed of RNA or DNA, that can bind to complementary sequences of RNA or DNA. For example, DNA primers are used to amplify DNA of interest in a PCR reaction, while RNA or DNA probes can be used in fluorescence in situ hybridization, in which one can look at specific DNA mutations or quantify number of copies of mRNA. This shows that many research tools are based on the same principles of recognizing biological molecules and investigating a myriad of structure and function in cells and tissues. Just to give a few examples of this, I showed 3 different tools based on the use of a fluorescent fusion proteins in cells. In the past years, it has been shown that fluorescence tools are very useful for investigating a variety of research questions. Especially the fluorescent protein fused to a protein of interest can shed light on many different cellular mechanisms. For example, in the first video, I use a GFP-tagged histone protein to investigate laser toxicity in live fruit fly embryos. In this video, you can appreciate the simultaneous nuclear divisions in this early stage embryo. In the middle, we used a GFP-tagged transcription factor of cartilage, SOX9, to investigate the transcription factor activity with a method called fluorescence recovery after photobleaching. In the third example, we used Foerster resonance energy transfer, or FRET, of cells expressing ERB1 EGFP and showed that multiple molecules of the EGF receptor aggregate in the presence of the ligand EGF, which is visible by higher fluorescence intensities and lower anisotropy values. Many more research questions and tools are available. When you plan your experiment, you need to make sure that you use the right tool for the question. You can learn a lot from reading papers and looking whether the right tool is chosen for the experiments described. Thank you for watching this video. If you have any questions, please email me or respond below this video. In these links you can find the original papers for the data shown. In the next lecture series, I will talk about computational modeling of signal transduction networks using ANIMO.